So I'm going to summarize for you, for those who didn't have the opportunity to attend the scientific meetings for the last day and a half. For those of you who did not attend, I would like to quickly summarize what transpired at that meeting, and then also kind of give you my own conclusions as to what the meeting brought us in terms of novel therapies for alpha-1 antitrypsin deficiency. So this is the title of this meeting, Novel Treatment Paradigms for Alpha-1 Antitrypsin Deficiency and Selected Other Conditions, and we'll hear about those selected other conditions as well. And there were three parts to this meeting. The first one was on basic observations. The second part, new treatments for lung and liver disease in alpha-1 antitrypsin deficiency. And then finally, new disease targets for alpha-1 antitrypsin as a therapeutic protein in individuals who are alpha-1 antitrypsin replete. So let's just start with the basic observations. The first presentation was made by Mike Iannotti, and he talked about high throughput liver cell model. This is a unique technology that's available at NCATS, which is a National Center for Advanced Translational Science at the NIH. And what he's able to do is develop a cell system which can tell us whether or not alpha-1 antitrypsin, the Z protein, accumulates in cells or gets secreted out of the cells. And then what you can do with this system, if you can determine that outcome, you can run a whole bunch of different chemicals through that system to determine which one might actually have a beneficial effect on that. And most of you probably know that the liver disease is related to a retention of the polymerized alpha-1 antitrypsin, which is an abnormal alpha-1 antitrypsin because of the gene defect. And these molecules, they kind of stick together and they accumulate in the liver cell, and that's toxic to the liver cell. So if you have an agent that can make this protein get out of the cell, you will protect the liver cell, and therefore the idea is that you will protect liver disease from occurring. So he has this system whereby he can actually look at this, and he has already run a high number of chemicals through this system and has a few hits which may be promising going forward. So this is one approach which I believe is going to be very successful going forward in trying to find new therapies for liver disease and maybe ultimately for lung disease as well. The second presentation was entitled New Therapeutic Targets for Alpha-1 Antitrypsin Deficiency, presented by David Lomas from England. And he was talking mainly about small molecules that could be used in the liver to prevent these molecules from sticking together. And if they don't stick together, they're probably not going to be that toxic to the liver cell, and it's easier for them to be secreted out of the liver cell. So he talked about that, and he has a lot of experience in doing research along those lines. And then he also found something very interesting, is that he has developed an antibody to Z-alpha-1 antitrypsin that is polymerized and circulates through the blood. So that could be used eventually as a biomarker for liver disease in alpha-1 antitrypsin deficiency. That's something that we don't have presently, and it could be helpful for clinical studies in the future. The next presentation was made by Andrew Wilson from Boston, and he's using iPS cells to study potential therapies. So these are cells taken, for instance, from the skin of patients who have alpha-1 antitrypsin deficiency, and these cells can be reprogrammed to become liver-like cells, or eventually maybe lung-like cells. So various cells in the lung could be reproduced like this as well. And then you can use these cells from a patient to actually look at the effects of drugs in vitro. So now you have made a liver cell out of a skin cell in a patient with alpha-1 antitrypsin deficiency. And if you're talking of personalized medicine, you cannot be more personalized than this. So this is an approach that we hope in the future will be very useful in finding new treatments for alpha-1 antitrypsin deficiency. They have also generated an iPS cell repository, and they offer this as a resource for investigators worldwide. As far as I know, Andrew, it's at no cost. This is really a great service for investigators who would like to use these cells. Then Bruce Trapnell talked about macrophages for gene delivery to the lung. Now, he was talking about a disease other than alpha-1 antitrypsin deficiency-related lung disease. It's a disease called pulmonary alveolar proteinosis, which is also a genetic disease, and it's caused by abnormal macrophage function. Macrophages are kind of inflammatory cells that are present in the lung, and they do a lot of different things. But one of them that they do abnormally in this particular condition is they don't remove proteins from the lung that are normally secreted. And that's why it's called alveolar proteinosis. And he actually has developed an intervention whereby he takes the macrophages from the patients, he transfects them. So he does gene therapy with the macrophages, puts the macrophages back into the lungs, and that will essentially cure the disease. He's now planning to do a human study, but so far this intervention has been very effective in a mouse model. So I think we have a good prospect here of having a new way of putting genes into the lung and therefore correct abnormalities, including eventually maybe alpha-1 antitrypsin deficiency, although we have no evidence for that as yet. And then Jeff Teckman talked about preclinical model studies for alpha-1 antitrypsin deficiency liver disease. He has a lot of experience in this. He has developed a mouse model for alpha-1 liver disease. And he kind of summarized all of the studies that have occurred already in the past using this mouse model to look at new interventions for liver disease in alpha-1 antitrypsin deficient individuals. So this is the end of the basic observations, and my conclusions were that we have now promising cell and animal models to assess the potential of interventions such as gene therapy, gene silencing, small molecules, etc., for subsequent use in humans. I believe that these early studies that have occurred in the research community in alpha-1 antitrypsin deficiency have been very useful to bring us to a point where you can now plan early phase studies in human subjects. And I'm going to talk about some of those in the next session, which I will summarize now. And that is new treatments for lung and liver disease. First, Dr. Gruntman from Boston talked about intramuscular gene therapy for alpha-1 antitrypsin deficiency-related lung disease. That it's at Mass U, they have a 15-year history of doing this. It started at the University of Florida and then continued up there. Their research has progressed from animals to humans. And they have found that if you transfect muscle, so you inject the normal gene, the MM gene, into muscle, the muscle can generate normal alpha-1 antitrypsin, which then hopefully will go into the circulation and do its protective function in the lung. They have been able to do this quite successfully in terms of having the expression of the gene in the muscle, which is very long-lasting. The only problem is that the serum level elevation that occurred with this intervention was relatively little. So it would be of no therapeutic consequence, I believe, at the present time. but they are working on further improvements for this particular intervention, and there's still a hope that in the future we will have a gene therapy, which in this case involves transfecting muscle cells, which then can produce normal alpha-1 antitrypsin and put it into the circulation. Another way of doing this is intrapleural gene therapy, and Ron Crystal presented the early data that they have in that. So far, he has tested this approach in mice and monkeys. And essentially what it involves is that you actually put the gene into the lining of the lung. It's called the pleura. So this is the lining of the lung inside the chest wall. And by doing that, by transfecting those cells with the MM gene, he has found that he can now find expression of that gene in the lung. He can find expression of that gene in the lining of the lung, but also in the liver, which means that he has a remote target for this particular approach as well. And he has found that in mice and monkeys so far. And he's now in collaboration with a biotech company in the process of planning an early phase human study. So this would involve for you as a patient, if you were to participate in such a study, is that they would puncture your chest wall and they would put this material into the space between the chest wall and the lung. And that's how the whole process would start. Very exciting, but again, at this stage, we only have data in mice and monkeys. Then, of course, we had the presentation on aerosol therapy for alpha-1 antitrypsin deficiency-related lung disease, and that was presented by Dr. Toth from Kamada. Kamada has sponsored 2 trials with this, and what they have done is they administered between either 80 or 160 milligrams of alpha-1 antitrypsin into the lungs by aerosol, and this is administered twice daily, this total dose. They have found in these studies that this procedure is safe. They have found that it increases the alpha-1 antitrypsin levels in the lung to a protected level. And they have also some indication that this intervention will slow the decline in lung function over time and also has other beneficial effects, such as effects on flares of COPD. It will reduce the number of those flares, and it may have an effect on some symptoms as well, such as shortness of breath. So this is very promising, and we hope that these early observation, which actually also involves a phase 3 study, will hold up as we go forward. So again, quite promising. And then here was a presentation by André Quentin, which was very interesting, and that is to look at antioxidant therapies for lung disease. So this would be non-specific, not just for alphas, but this would also be useful in patients with common COPD. And the notion is that cigarette smoke, either directly or by producing inflammation in the lung, produces oxidants, and these oxidants can be toxic and do damage in the lung, so-called oxidant damage. And the idea is that if you come up with certain treatments that will be treatments that promote antioxidant activities, you could actually have a beneficial effect in patients with COPD. And he talked about broccoli. He talked about selenium. He talked about a special type of prostaglandin and superoxide dismutase. So the last 2 would be drugs. The first one is diet. And the second would be supplementing selenium in individuals who have a selenium deficiency. So selenium doesn't really work if you have normal selenium in your body. But there are certain regions in the world, I understand, where there is selenium deficiency. So strong recommendation to you to eat as much broccoli as possible. And he also indicated that you have to make sure that some of the stem is still there. So you cannot just clean everything out and just take the green stuff. You have to do as much of the broccoli plant as possible to have the maximum effect. And then Dr. Hamilton presented data on silencing RNA for liver disease. And now what this does is this is an intervention where he uses RNA to shut down the Z protein production in the liver. So this is really a very basic intervention, because you say if you don't make abnormal protein in the liver, it doesn't polymerize, you will reduce liver toxicity. So it's a very smart way of thinking about this. And they have actually found that this intervention drastically reduces alpha-1 antitrypsin levels in serum, which means that now the liver has been shut down, so the protein doesn't come out of the liver because it's not being made. So this is one way of looking at this. The other one would be to do a liver biopsy. Now, they haven't really gotten that far yet because in some toxicity studies, it appears that this kind of intervention can cause death in monkeys. So this was shut down at the present time, this study. It was actually shut down by the FDA. And the good news, however, is that according to Dr. Hamilton, Arrowhead is still interested in pursuing this approach using a different method of drug delivery to the cells. So he believes that it is not necessarily the RNA, the silencing RNA, that is the problem, that it may be actually the chemical with which the RNA is fused that can cause this particular problem. So we are still hoping that this will go forward. Similarly, Dr. Haslett from a different company talked to us about a different way of getting the silencing RNA into the liver cells. And they found in early human studies that it, again, lowers serum alpha-1 antitrypsin levels up to 6 months or even longer. But they found a transient liver toxicity in a few of the healthy volunteers in whom they tested this drug first, and they decided to stop the study because of that. So this was not mandated by the FDA in your case, but they unilaterally decided to stop and not go forward with this approach. But again, in their hands, they don't want to shut down this program. They are still planning to go ahead with it in the future by modifying some of the methodology that you're using. So here are my conclusions for this part of the meeting. We have extrapulmonary gene therapy and gene silencing, which are closer to becoming a reality in human alpha-1 antitrypsin deficiency. We have now encountered, at least for the liver disease, some bumps in the road, but I believe that's all they are. Aerosol alpha-1 antitrypsin may become an alternative to intravenous alpha-1 antitrypsin in the future. So I think this is really something that we should take seriously at the present time. And of course, don't forget to eat broccoli. Now, the last session talked about using alpha-1 antitrypsin as a therapeutic protein for diseases that are not associated with alpha-1 antitrypsin deficiency. And the reason for this is, and that was pretty much explained to us by Dr. Lewis, and then we had two additional presentations, one by Dr. Song and one by Dr. Shapiro along these lines. But the idea here is that alpha-1 antitrypsin has a broad anti-inflammatory and immunomodulatory effect. So you can think of using this for diseases other than alpha-1 antitrypsin deficiency. In alpha-1 antitrypsin deficiency, we're using it to replete a deficient protein. In this case, we are going beyond and above the normal level to have beneficial effects in other diseases. And they presented data which, first of all, what I would like to say is that based on this expectation, there are several diseases where basic research has indicated that alpha-1 antitrypsin could be beneficial. And those conditions are, believe it or not, as wide-ranging as type 1 diabetes, systemic lupus erythematosus, organ transplant rejection, graft versus host disease, influenza infection, cystic fibrosis. So you have a wide range of different diseases. These have nothing to do with alpha-1 antitrypsin deficiency, and yet alpha-1 antitrypsin could be a therapeutic agent in these conditions. Several ongoing human protocols were reported, and a new term was created. Not alpha-1 antitrypsin deficiency, but alpha-1 antitrypsin insufficiency, where the alpha-1 antitrypsin doesn't rise high enough in these various conditions above normal to actually have its beneficial effect. So therefore, you have to give it therapeutically. So my overall conclusions are as follows. Gene therapy, gene silencing, and small molecules for alpha-1 antitrypsin deficiency, liver and lung disease continue to have a future. Early human trials are likely to continue. Aerosol alpha-1 antitrypsin is safe and could be effective in alpha-1 antitrypsin deficiency-related lung disease. New disease targets for alpha-1 antitrypsin will remain an interest for basic and clinical investigators. And I would say that although the title of this meeting was Novel Therapies, I would say we may not have novel therapies yet, but we certainly have emerging therapies. And that's very encouraging to me. So we have emerging new therapies and optimistically can expect novel therapies for alpha-1 antitrypsin deficiency, liver and lung disease in the foreseeable future. This was the third International Research Conference on alpha-1 antitrypsin deficiency. And I believe that the topic of this one was very close to patients because we only talked about therapies. And ultimately, that's what everybody wants. So we are close. We are close to novel interventions. We are not quite there yet. And we are slowly getting closer to a cure for alpha-1 antitrypsin deficiency. So I think this was a very successful meeting. I would like to thank Dr. Jerry McElvaney, who was the chair of the meeting. We had all the experts we needed in the room, and there was very enthusiastic discussion that took us through the meeting, and we look forward to the publication which we will have, which will actually summarize all the proceedings for everybody to read. So thank you for listening, and for those of you who were there yesterday, I hope that this summary reflected what actually transpired there. Thank you.